Welcome back, everyone. This is the fifth of this year's Burlington House Lunchtime Scientist Talks. We had a break last week with half term, but we are back with a new set of speakers for June. My name is Lucinda, and I work at the Royal Astronomical Society, which encourages the study of astronomy, solar system science, geophysics, and other closely related branches of science.
Yes, hello. I am Joe, and I'm just a voice at the moment because Lucinda needs to turn on my camera. But it is great to be back for this series of talks about astrobiology. Yeah, as I said, I'm Joe and I work at the Linnean Society, which is a natural history organization which aims to inform, involve, and inspire people about nature and its significance. We have really special collections in our building. We have amazing programs, learning programs, and we also have 3 scientific publications.
You should be able to turn your camera on now, Joe. Sorry about that.
That's okay.
And a little bit more about our collaboration. So the Royal Astronomical Society and the Linnean Society both live in Burlington House, a great big building in the center of London, alongside other cultural organizations such as the Royal Academy of Art, the Royal Society of Chemistry, the Geological Society, and the Society of Antiquaries. The RAS and the Linnean Society have co-produced this series of for young people with an interest in astrobiology to learn about this exciting field and the variety of careers that people can have.
Yes, this webinar is taking place on Zoom if you're on Zoom, but we're also streaming onto 2 different YouTube channels. So you might be watching us on there or on a different social media platform where it might be posted. If you are watching this live and not sometime in the future, you can type questions into the Q&A box on Zoom or onto either of the chat boxes on YouTube. Our speaker will try and answer as many of them at the end. We will put those questions to them if we can. Our speaker today is Professor Willie Wilson, who is the director of the Marine Biological Association. In particular, Professor Wilson is a marine virologist who studies and specializes in algae and viruses in algae. Willie's personal research interests focus on the many roles of marine viruses, which he's quoted as describing as the lubricants of great engines of planetary control. We've heard a little bit about microbiology and space in this series, but nothing on viruses yet. So I think Lucinda and I are very excited to hear more about ideas around astrovirology. So thank you very much for joining us, Willie. Can I hand over to you now?
Excellent. Thank you very much, Joe. And thank you, Joe and Lucinda for for the invite to this. I think when I first received the invite, I did what any good self-respecting scientist does and went on to Google to find out what astrovirology actually was. So I think it's quite interesting that the more I read into it, the more I realized that I was an astrobiologist with the type of work that I do. So what I'm going to do is share my screen here and get the talk up and running. Now, I guess it is a fascinating subject. I mean, I'm actually a virologist and I'm a marine virologist, and I think I hope that's not too disappointing for most of the audience here, but thinking that you might be sort of learning about viruses in space. So I guess part of the punchline here is that I'm not going to tell you about viruses in space because, quite frankly, we don't know about any evidence of viruses in space. But astrobiology, as I'm sure anybody who's been sort of looking at some of the astrobiology talks, it's about it's really about exploring the art of the possible, I suppose. I mean, what if you were in a planet in one of the Goldilocks zones, and you sort of traveled about a few million light years in your spacecraft and came over to planet Earth looking for life. Chances are you would take your little test tube and you'd probably get a sample of seawater, because seawater, of course, covers about 3/4 of the planet in and the oceans that we have. And if you had a look in there, what you will find is viruses. Is going to be dominated by viruses, and that's really what I'm going to talk about in the talk today. And then I'm going to try and flip that a little bit by saying, well, okay, so that if this was an extraterrestrial from a Goldilocks zone planet, they come here, what would we need to do if we were going to go to another planet somewhere and look for evidence of life? And I guess the overall hypothesis that I have is that if you're going to look for life, you should be looking for viruses, because without viruses, you won't have life. And that might be a little bit surprising to some people. So I'll get a bit of a health warning here with as I go through this. I'm experimenting with a soundtrack that we seem to get it working at the start when we were practicing this, and I've got an animation, so hopefully all that's going to work. So, well, let's just start with the very basics. What is a virus? Well, I think it's a little bit topical at the moment. I think everybody should have heard about viruses by now. This is, of course, the coronaviruses all over the news, where it's caused sort of huge devastation, really, socioeconomic problems, and the kind of worldwide pandemic that it's caused, and it seems to be at the forefront of everybody's minds across the entire planet right now. And I guess part of the story that I tell is really that viruses are necessary for life, and without viruses you wouldn't have life. And probably, if you're being infected by something like coronavirus or some of the many other sort of horrible infectious human viruses that are out there, you're probably not going to want to hear that kind of positive story that comes with viruses on a planetary dimension. So I think there's a complete flip side to it, because the reality is there's these types of viruses like coronaviruses, Ebola viruses, flu viruses, all your human types of viruses are just a tiny fraction of all the viruses that you will find on the planet. So let's just get to the what even is a virus. Now, I think one thing I would recommend is that if you go onto YouTube and just type in virus animation, there's a plethora of brilliant videos and animations of virus infections. There's some very talented artists out there who've really made this very accessible. But in essence, all a virus is a very simple piece of biology. You have a nucleic acid, either RNA or DNA, a tiny piece of DNA or RNA, wrapped in a protein coat. Sometimes there's lipids on the outside of it. And in essence, they are parasites. They will attach to a host cell, hijack host replication machinery, produce multiple copies of itself, kill the cell, burst out, releasing new progeny viruses. And you get, and if I can show that's what I've just described there, is a kind of lytic type infection, which is a classic infection. These are your lunar landing module type bacteriophage that you get in these types of infections. These are viruses that infect bacteria. But you got, this is that's a kind of what's termed a lytic virus. But you also get a huge number of different types of what they term persistent type infections, such as chronic infections, where the virus can infect a cell, but the cell just kind of gets sick, if you like, and it just produces sort of small amounts of virus, continually produces small amounts of viruses. And then you go to the other end of this spectrum, where you get almost what's termed hidden infections. lysogenic infection, which is a term used in bacteria, or latent infections, where you can actually have, and all of us watching this, we're as human beings, we're going to be full of a huge different number of and diversity of latent infections. And nowadays, when you do all the genome sequencing of every organism that you can find, you're always going to find remnants of virus genomes within there. And it's not really known the function of a lot of them. They say they're hidden infections. If the conditions are right, they might then sort of burst out and sort of turn into either a chronic infection or a lytic infection. But they are pervasive throughout life. So let's move into, so when I was doing a little bit of research into this, I did a project a number of years ago, sort of looking at, this is sort of working with the University of Nebraska over in the US, we were actually developing a comic book, in fact, where we were trying to find virus superheroes. So this was actually really trying to get sort of looking at the planetary realms within the universe, and trying to get the idea of, well, if you had an ocean full of viruses, are they the superhero or are they the villain? And so what we ended up doing was saying, well, the villain is the organisms that they infect. In this case, this sort of monster-looking thing here is an algae that's something we call the Millenia huxley, and I'll go on and talk a little bit about that. And I guess that, at the time there was a lot of news around about algae blooms that were causing problems. Now, I'm a little bit uncomfortable with that analogy, because algae on the whole are they fix carbon dioxide and start of the food webs, and so they're actually very good. We need the algae, but we kind of go on to say that within the comic book. And of course, we have our superhero here, this sort of cool looking hunky bloke here, who's a superhero called EHV. And for geeks like me, EHV is a Millenia huxley virus, and he's sort of saying it's okay, just because I'm a virus doesn't mean you got to make that awful grossed out face. I'm one of the good guys, sort of real life aquatic superhero, and cool virus. We're really trying to make viruses seem a bit cooler than they get a lot of bad press, so we're going to try and make them look a little bit cool. So cool viruses like me possess the superpowers to combat the growing menace of a Millenia huxley. I'll talk a little bit about that later. Restoring the ocean to its natural balance. That's kind of the punchline here that we were trying to get across with this, is that, for whatever planet that you're in, is that the viruses are there trying to restore the balance. They act as what I call the lubricants of planetary control. And I'll come on and talk about that in a little bit. Now, this is the kind of image that you're going to be used to. So, in the Royal Astronomical Society, sort of looking at, this is a great image from, like, the NASA. When I was looking for these types of images, and I sort of looked at this and realized, well, I kind of look at images like this all the time. If you were to come from one of these galaxies, like I said earlier on at the start of the talk, and come in and look, and you had the right tools, and took your test tube of water away with you and had a look in it, you would actually see something like this, which actually, there'll be a few of you out there trying to guess what the constellation is here, but actually what this is, we've taken a seawater sample and added a nucleic acid stain that makes it fluoresce bright green, and then looked at it down a microscope. So we've gone from astronomical scale right down to microscopic scale. And every one of these tiny stars that you see there in this image is actually a virus. And the brighter kind of planets or galaxies that you can see are actually microbial organisms. These are mainly bacteria. And so that extraterrestrial coming to planet Earth is going to come in and have a look at this. And one of the things they'll find once they do the calculations is that on average, there's anywhere between 1 to 100 million viruses in a milliliter of seawater. Just take a moment to just think about that. I mean, if I was to go out in front of my office in Plymouth Sound and South Coast and out in the English Channel and took a milliliter of water, there's probably going to be in the region of about 10 million viruses in a milliliter of seawater. It's a huge number of viruses. And it's not just because Plymouth Sound is contaminated or really bad. That's just a kind of naturally occurring viruses. And we can get some fun statistics with that. If you sort of multiply that up, we kind of estimated there's around 10 to the 31 viruses in the ocean. We know there's around Avogadro's number, which is about 10 to the 23 infections every second. If we were to weigh all these viruses, it'd be equivalent in weight of about 75 million blue whales. Now, which is a massive amount. I mean, there's only around, there's less than 10,000 blue whales in the ocean at the moment. If you were to put all these viruses end to end, each of these viruses about 100 nanometers, they would reach about 42 million light years. Now, our own galaxy is only 100,000 light years. So you're sort of looking at that, all these viruses stretching, between 40 to 60 galaxies away. But the key message here is when you, if this extraterrestrial was to come and sample, most of what they're going to sample is microbial life. And these viruses are necessary for that life, for the recycling of the nutrients, for the transfer of genes through horizontal gene transfer. And what is that life even going to look like? I mean, it has, if you were to take your sample of seawater, who are all these viruses infecting? It is microbial life. These are some of the microbial animals in the plankton that are going to be out there. Most of these are animals. You have actually, there's a plant here. This is something called a diatom. There's a whole range of sort of weird and wonderful extraterrestrial looking animals, and certainly in a lot of your space movies, they take inspiration from a lot of these sort of planktonic organisms for some of the extraterrestrials and monsters that they create within the movies. You go down the scale, sort of looking at some of the plant microbes that are out there. This is something called a Miliana huxley, as a small single cell photosynthetic single organism that's about 5 microns in diameter, and we've got one on the end of a pinhead here, obviously photoshopped, but and for the really geeky ones out there, you probably have spotted the deliberate mistake already, but there's that cell is actually massive because a pinhead is about 75 microns across, so you could probably fit about 15 of these cells across at the top of a pinhead like that. But the point here is that most of the life that you're going to find throughout the planet, if you're going to be taking a random sample, is going to be microbial. You're going to have your phytoplankton, which are your plants, the microbial plants, and you have your microbial animals as well, the zooplankton, and then you of course you have all your bacteria, your archaea, that are going to be that actually, in fact, your bacteria account for around 90% of the biomass, pretty much anywhere on the planet. And for all this life that's there, you're going to have at least 10 different types of viruses that will infect each and every organism out there. So very clearly, if there's that many viruses out there, they're going to be having an effect on controlling the life, controlling the nutrients. I've got in here the dissolved organic material that help with the sort of recycling of the nutrients that are out there. And viruses are instrumental in all of these different processes. And if you take a look at our planet, of course, it's called the blue planet. And there's a huge amount of sort of microbial biomass is in there. And I always like using this slide, and there's a great quote that I always use in a lot of my different talks, is that the illuminated region is only a small part of the 3.7 kilometer mean depth of the ocean, yet it houses several of the great engines of planetary control. Now, two of the big engines are the absorption of heat energy, which drive a lot of the physical processes in the ocean. But then there's the absorption of light by the primarily single-cell phytoplankton, single-cell plants that essentially drive the biology in the ocean. And viruses are going to be, I like to term it, sort of using the engine analogy, are the lubricants of the great engines of planetary control. Without viruses, the whole system will just seize up. So, like I said, the hypothesis here is really that if you're going to be looking for life in any kind of planetary system, you should be looking for viruses. And as just to try and sort of make that point, we're going to sort of look at an individual case study. This is a system we've worked with for a number of years, over 20 years now. Something called the Meliana huxleyi. That's the one I showed on the pinhead. It's a single cell phytoplankton, a single cell plant in essence, microalgae, and it's infected by actually a giant in virus terms. It's about the size of a tiny bacteria. Actually, the virus itself is about 200 nanometers in diameter. And so what I'm going to do, I'm going to have a break now because, and I'm going to try a little experiment where I was involved in a podcast, a radio podcast. Probably, it must have been quite a few years now. I was actually still working in the United States at the time. And we put a podcast together. Now, one of the things I'm going to say is that the podcast is the professional bit, because that was done by a professional radio company in the US called Radio Lab. The slides that are cobbled together to go along with it were done by me, and there's quite a nice animation in there as well. And what it really does is it explains the virus infections, how they work, and then sort of looking at it at a kind of planetary level. So what I'm going to do, I'm just going to, don't worry, I've rehearsed this. I'm going to put on a soundtrack. Hopefully I can get that working. And Joe will come in if it isn't working. So I'm just going to get that started.
All right.
You're
listening
to Radio Lab.
Radio Lab.
Shorts! From
WNYC
and NPR.
I'm Robert Krulwich. This is Radio Lab, the podcast. And today we're going to talk about global warfare. A vast battle across the planet on a scale that is really hard to believe, involving trillions of deaths. And yet, we really need this war, because without it, I wouldn't be here, you wouldn't be here, Jad wouldn't be here, and you may have noticed he isn't here, and it's not because of a war, it's because he had a baby. But before Jad went on paternity leave, we sat down with Ari Daniel Shapiro.
Okay.
And he told us this story.
Here it comes.
Oh, right, yeah. So here we are at the Center for the Culture of Marine Phytoplankton.
This is Willie.
And I always say
Willie Wilson.
Willie Wilson is his name?
Yeah.
That's tough.
Just like his grandfather, actually.
Yeah. My dad's William Wilson. There's a long line of William Wilsons.
And your son?
My son is Angus, but he's Angus William Wilson. So
I love that guy.
Willie works at the Bigelow Laboratory for Ocean Sciences in Maine.
Okay.
And he studies these tiny plant-like creatures that live in the sea called phytoplankton.
right.
He keeps them in a fridge in these little test tubes, half full of kind of greenish water.
Let's have a look at that.
He pulled one out and showed it to me. And it doesn't really look like there's that much going on in there.
But actually, right at the bottom, you can see what looks like a
White.
sort of goo.
What is it?
It's the carnage of war.
Oh.
In that test tube that Willie's holding are millions of tiny single-celled plants called coccolithophores.
Coccolithophores.
Yeah, and there are lots of them in the sea.
There's probably about 100,000 of these coccolithophores in a teaspoon of seawater.
Tell me about the coccolith. Like, what do they look like?
They're basically like tiny little translucent balls with, there's a slight tinge of green. But the key thing is
the outside of that ball, it has these white plates.
Tiny circular shields of chalk.
Because the coccolithophores are fighting for their lives.
Fighting with whom? Each other?
Viruses. These viruses that are shaped like diamonds. So here's what happens. Imagine you're a coccolithophore, floating in the ocean, and along comes this diamond-shaped virus. And it jams its diamond tip into you.
Between these plates and actually get inside the cell.
The chinks in the armor.
That's right, it's like the chinks in the armor.
And the coccolithophore just engulfs it.
And the virus thinks, yes, I'm in here. And then it sort of makes straight to the nucleus.
And it's at that moment that the viral takeover begins. The virus kind of hijacks the cellular machinery that's usually used by the coccolithophore to make more coccolithophore stuff. And it starts making more viruses.
So inside the coccolithophore now, there are these little diamonds multiplying.
Yeah, they're filling up that space. And eventually, all these viruses head out of the coccolithophore.
In big belches or?
Like a steady stream of viruses.
Wow.
And each one of these viruses has the ability to go on and infect another coccolithophore cell.
In fact, those coccolithophores in that test tube that Willie showed me,
if I shake this stuck to the bottom a little bit, yeah.
Those were in the process of dying.
You smell that? Yeah, so that, what you're smelling there, that's the infection. That's dimethyl sulfide you're smelling. So the infection is already occurring in this culture.
And when the coccolithophore dies, those white shields kind of fall off the cell.
They sort of gradually sort of rain off over the course of the infection.
So as it's dying, after it's spewed out these viruses, it just sheds its plate, and it kind of, and then it dies.
Yeah.
And that creates this white chalkiness.
Yeah.
Well, so this means like the coccolithophores are not doing very well.
Well, they've got a couple of tricks up their little calcified sleeves. Sometimes when a virus enters, the coccolithophore will send out a chemical signal.
They're sort of shouting, hey, it's too late for me.
But save yourselves.
Oh.
And initially, this signal's pretty weak in the water. But as more and more coccolithophores are infected, the chorus of this chemical beacon grows louder and louder.
And so the other cells, they hear these messages.
And they change by messing with their DNA a bit. And they go from having those white shields on the outside to having these jaggedy scales.
Which we think might be impenetrable.
Scales instead of these plates.
That's right, yeah.
Shields.
Yeah, that's right.
Why aren't they just scaly all the time?
because when they're scaly, they can't be the best coccolithophores they can be. They just don't grow as well.
So scaly is an adaptation against the viruses.
Exactly. And then finally, if all else fails,
Programmed cell death.
the coccolithophores just commit suicide.
It just shuts down and kills itself to prevent propagation of viruses.
But over time, the viruses have figured out how to
Prevent the cell from killing itself.
So it delays the death of the coccolithophore for as long as possible to maximize the number of viruses that can get out.
Wow, this is serious.
Yeah, it's like an arms race.
There's a constant battle to be fitter than you were several generations ago. And without...
Here's the crazy thing. This battle is happening all through the surface of the ocean. There are legions of coccolithophores dying all the time. And the coccolithophores are shedding their white shields.
It's like taking millions of tiny little mirrors and putting them in the surface of the ocean.
So many that you can actually see this carnage from space.
You can see this from space?
Yeah.
You get the massive blooms that cover almost the whole of the North Atlantic. I mean, you get this sort of milky bloom that covers anything from, off the west coast of Scotland under southern Iceland, almost all the way to Newfoundland. In the southern hemisphere, you get this sort of massive milkiness that circumnavigates the globe.
These vast swirls of milky water curling around islands and continents.
And that's all carnage from this battle.
Billions and billions of soldiers that have fallen in the field.
Wow.
That we can view from space.
It probably is trillions if you're talking on that scale.
Yeah, I think,
What's after trillions?
Quadrillion?
You think it's we're in the quadrillions? Just say it, see how it feels.
There are quadrillions of soldiers dying.
How did that feel?
It felt good. If I were to be an astronaut, how often would I see these sorts of blooms?
All the time, somewhere on the planet.
Every day, every hour?
Every day, every hour. There's going to be a bloom going on somewhere. A good example is off the Norwegian fjords. They start in the fjords in late April into May time, and then they sort of creep out of the fjords like this huge living amoeba. Fishermen hate it because the fish can't see the lures, so they can't catch the fish anymore.
And as the shields rain off and fall down to the ocean floor, they build up and build up over time.
Millions of years of sedimentation of these sort of chalk particles.
That's actually what led to the creation of the Cliffs of Dover, the White Cliffs of Dover in England.
Shut up, really?
Yes.
This is sort of geology in action.
And not just that, when the coccolithophores get decimated by the virus, it kind of clears out the ocean for other phytoplankton to bloom. And then they get mowed down by their viruses. And then the coccolithophores might bloom again, and then they get wiped out. And this cycle...
all these battles, it's all responsible for about half the oxygen that we breathe.
Really? Half the oxygen we breathe?
Half the oxygen. Whoa. Because when the phytoplankton bloom, they take in carbon dioxide and they release a puff of oxygen. And then they're cut down by these viruses, but they grow back up again, and another breath is released. So the whole system is just kind of breathing.
People think of the lungs of the planet are the rainforests, and that's kind of half the picture. But every other breath we take comes from the phytoplankton in the ocean that are going through these battles on a daily basis.
So this is a battle that rages every single day somewhere in our oceans.
Yeah, we need the battle to live.
We can hear you now, Professor.
Oh, you can hear me now, right? Okay, thanks. Let me get back to here. Right. I need that. Okay. So what? Yeah. So what that really demonstrates is just a kind of planetary level. Oops. I've been asked to start my video. I think I do that. Yeah. Okay. But I mean, certainly, I mean, if we were to sort of flip all this, so essentially what we've said, the whole point of what I've just been talking about over all of that is that at the planetary level, the viruses really are necessary for life. But if you were to go to a different planet, perhaps, I mean, I've talked about the Goldilocks zone planets, but of course, there's a lot of the planets are going to be in extreme conditions. And this guy here, this is Ken Steadman. He's a good friend, colleague of mine. He's I guess he's what I would call a true astrovirologist, and he studies viruses in extreme environments. And here he is in the Rabbit Creek in the Yellowstone National Park on a kind of average day for Ken, where he's collecting samples from water that's anywhere up to about 90 degrees centigrade, really acidic pH way down to 3.5. And he spends a lot of time sort of looking at viruses in extreme environments. And I would certainly encourage you to sort of look Ken up. And he gave me a couple of slides here. This was the other one that just shows electron micrographs of a whole range of the different viruses that you can find in these extreme environments, and they're infecting a group called archaebacteria, which are ancient bacteria that have where kind of life started on our own planet. And it's likely that if you're going to go to some of the planets that are much closer to our own galaxy, where you have these extreme environments, then it's probably going to be something like archaebacteria that'll be living there, and these are going to be the viruses that you'll find that infect them. And one of the things that Ken has found is that they actually, some of these viruses coat themselves in silica, which could be a way of protecting them. And he's done a whole range of experiments where, after they've been coated in silica, they can still remain infective. They can survive for sort of long periods of time. But a large part of what we need to be able to do if we are going to be exploring other planets is actually looking for these biosignatures. And a lot of work that Ken does is sort of looking for the biosignatures, sort of looking for viruses coated in silica, and perhaps even so looking at all the different shapes, looking for these sort of weird and wonderful shapes of the viruses might be some kind of biosignature for these different viruses. And a lot of the work that we've done within the ocean, I mean, we use a device called a flow cytometer, where essentially you take a stream of water and you pass it by a laser. Now, if you think back to that, the slide I had with all the tiny green dots on it, what the flow cytometer will do is pick up these green dots, and where you can find, it creates a signature like this. You can look at a side scatter of each of these virus particles and plot it against the fluorescence. The side scatter is just a sense of the size of the virus, and you plot it against the fluorescence. And you can see all these different groups that come up in the signature. And this was we were looking for giant viruses within this experiment here. Bacteria are way up here, and you can actually sort them. You can actually pull out all these individual viruses and get single viruses. And we have what we call our virus toolbox. I give lots of different sort of extended talks on this, where we can actually look, the biosignature we're looking for is the genome or the DNA of the virus. So if you can see the virus on the flow cytometer, and here we have another signature where we're looking for virus-infected cells, and then you can actually sort out these at the single level, single virus level or single cell level. And then you very simply go through a program of sequencing. We go through a process called multiple displacement amplification, where you can amplify up the very tiny amounts of genetic material that you have, and then you go and sequence, and then you put the sequence data through databases, and you can come up with, you can put all the sequence information together. I mean, we do it in pie charts like this. And then you can do a taxonomy by comparing that sequence to sequences in databases. And what we find is we just come up with thousands and thousands of sort of novel viruses that have never been sequenced before. So it seems, I mean, we want to go to other planets to sort of try and find life, but there's a lot of the arguments we make is that, well, we don't know the vast majority of the microbial life that's already within our own planet, particularly when it comes to viruses. I know I can go out in front of my office in Plymouth Sound and take a test tube of water, and I'll be able to isolate a whole range of viruses that have just been never been seen before, new to science. So it's a huge area to explore. So I'm going to bring the talk to a close now. The astrovirology. I have to say, it has been a learning experience for me. I've really enjoyed doing a bit of research on this. I sort of realized that I think I must be an astrovirologist with just the way that we kind of think about virology and the tools that we'll require if we do need to go to other planets. I've talked about, we've got astronomical virus statistics on planet Earth. Wherever there is life, there's viruses are going to be found in abundance. You need viruses for life. We know the viruses thrive in extreme environments. So this says, well, that there could be viruses in some of the more extreme conditions that you find in some of the other planets. The challenges that we have are sort of really developing the tools for detecting the virus biosignatures. Now, whether that is for detecting nucleic acids or for viruses that in, for example, extreme environments, with the work that Ken's doing, sort of looking at viruses associated with silica, for example, and they would need to be miniaturized, that kind of technology adapted for space travel. But I would argue that viruses are the true extraterrestrials to actually lubricate planetary engines. So I shall just leave it there and open it up for questions. I'm thinking I've gone a little bit over what I was meant to, but I'll just stop there.
No,
shall I stop sharing my screen?
It's been great. Yeah, or you can take it off, whatever you'd like. So that's fantastic. So sorry, Joe, go ahead.
Yeah, I just wanted to say thank you so much for that talk. I find the world of viruses really interesting. I guess particularly because my whole understanding growing up and still today is that viruses are bad. I mean, they infect things, they cause disease, and they take over. I mean, the animation of them taking over the coccolithophores is intense. And so it's a challenge to my understanding and perception of this whole kingdom of things when we talk about good viruses and how integral they are to a whole ecosystem. I guess I feel like them in the same way as wasps, in that I know that they are really important for an ecosystem. And that's something that's changing over time, where we're starting to see wasps as things that we shouldn't just kill quickly because they might hurt us at some stage. Yeah.
And it's because we anthropogenicize everything. We sort of relate it back to ourselves, which is only natural to do. But I'm a huge fan of you get your Gaia hypothesis, where the planet is this self-regulating organism that everything's all interconnected, and that's when you sort of think of the microbial world and viruses in particular that are absolutely necessary to make sure that we have an ecosystem that humans can survive in.
Yeah. Do you think that, I mean, I'm not sure about wasps. People are doing great work on wasps, but I think bacteria has come a long way, our understanding and appreciation of bacteria. We talk about the gut microbiome and we're like, yes, we really need this. Have more yogurts. Do you think viruses are going that way, or has COVID kind of really put us on the back foot with the appreciation of viruses?
Well, no, I think you mentioned the gut microbiome. I think that the average person has about 1 kilogram of microbes, and for every single microbe there's a whole range of viruses that are infecting there. So there's a gut virome as well. There's a huge amount of work actually done on that, and it does just as much for well-being and personalities as has ever been understood before. So yeah, I think ultimately, when you get something like coronavirus, it can take over. It's a new virus that comes into the system. But if you sort of take a step back from it and sort of look at, when you have so many people on the planet and you get new viruses, and emerging diseases like this that come into the system, and of course as human beings we can adapt to that. I mean, there's by producing the vaccines that get it under control. It's this constant battle. I think we mentioned that in the talk there. So we have a way of getting around it. Nature has a way of getting around it. As human beings, our immune systems are adapting all the time. You hear about, early on they were talking about, well, just let herd immunity take care of it. Well, that was never going to solve it immediately. But if nothing had been done, if science hadn't come in and sort of helped develop the vaccines, just to accelerate what would be a natural process, nature has a way of sort of helping all the time like that. But yeah, I think that viruses, we just know so little about them in the natural environments. When I started my PhD, 30 years ago, we knew virtually nothing. In fact, just before I started my PhD, it was thought there was very few viruses, even in the ocean. And it was when people started looking down an electron microscope and saying, well, you know what? There's actually a huge amount there. Clearly, if there's that many viruses there, biological oceanographers were thinking, well, if you've got 100 million viruses in a teaspoon of seawater, they must be doing something. The planet, in a lot of cases, the ocean is very healthy. So they can't be having a negative effect. They must be having a positive effect.
Yeah. Lucinda, did you want to ask a question?
Yeah, there are some questions in the queue here for Zoom, but I'm a human space exploration fan and advocate. That's kind of what I've been working for the past over 20 years now. So, it's always been, being afraid of space exploration for, of course, microbes and viruses, and I've always been told, where there's where there's water, there's life. And so you're talking about this amount of viruses in water. I'm just going to ask a simple question here. Are viruses alive? Are they considered life?
Oh, that's a great. Okay, how much longer have we got? We have this debate. Yeah, we have this debate all the time. It's brilliant. I love that question. Are they allowed? So, you know what? I was almost going to get into that in this talk, but I realized I never had enough time. I mean, I've seen a great analogy for if you think of viruses as in itself. Okay, right. One of the questions I quite often give people who come and sort of want to have a job with me is, what's the difference between a virus and a bacteria? Now, a bacteria has inherent metabolism. You can take a single bacteria, you put it in some growth medium, and it'll divide. Okay, you take a single virus, and with that bacteria, and you'll get 1 million bacteria within a few weeks, a few days in some cases. You take a single virus, you put it in the pot, it's still going to be a single virus, years later, because it has no inherent metabolism. So it's in that respect, and it depends how it depends how you define life as well, of course. But if you take a virus and add it to a cell, and it starts dividing, okay, so. And then I have a great slide that shows a term we call a virial cell, which is a virus-infected cell. And then you're producing new viruses. So that in itself is life. And you can get really geeky about this in terms of sort of looking at the difference between a virus and a virion. A virion is the virus particle itself, which has no inherent metabolism and can't divide by itself. If you take the virion that infects the cell, it then is termed the virus, which can then divide. It's a little bit like if you take an acorn on its own. An acorn, once you give it all the right ingredients to allow it to thrive, it becomes an oak tree. So is the acorn an oak tree? It's the same kind of thing. It's like if you take a sperm cell on its own, it's not going to be able to divide. But as soon as it attaches to an egg, then you get the division. So it's a slightly different way of looking at it. Yeah. It is an important part of life, but in itself, I like to use the term, and you might have noticed in the talk, I use the term if it's infectious or not. If it's infectious, once it becomes infected, that is life. When a cell becomes infected, it's a different form of life. Yeah. And another way I've seen it described is there's only two forms of life, and that is non-infected cells and infected cells. So, yeah,
I'm curious to also know, we generally think of viruses as a bad thing, negative thing. Are there viruses out there that help us? You're calling them the lubricant for planets, that makes it go or makes it function. So it's a necessary thing. But are there viruses out there that actually benefit human beings?
Oh, yeah. So actually, one of the other speakers that you had talked about, bringing on Martha Clokey, who's a good friend, colleague of mine. And if you look up, so she's Professor Martha Clokey at the University of Leicester, and she does a huge amount of work using viruses as natural antibiotics. So you can so if you have a bacterial infection, and actually their natural enemies can be viruses, and it was used by probably 50, 60 years ago. It was quite common in Eastern Europe, where viruses that infect bacteria, bacteriophages, were used as a natural antibiotic to prevent stomach infections, and so that's been kind of quite well known. But they're also used. You can use them as sort of vectors for doing gene therapy work as well. So there's lots of different examples, because for looking at function of different genes, and you can use sort of viruses to insert into different genes to knock them out, to sort of work out what happens when that gene doesn't work anymore. So there's lots of really good examples of that. They're often used as tools in molecular biology.
Yeah, they sound like they actually have, there's a lot of benefits that are very helpful to human beings. That's really good to know and to discuss, or to mention anyway. I'm just going to ask one question and I'm going to hand it back to Joe. We might go a little long because we do want to ask you about the career paths and things like that, so that our GCSE students get an idea of how you got to here. But Carlo asks on Zoom, is it safe to say that viruses can be a precursor for a life to form on a planet? Like they need to be there first before life to evolve? Or is it a big question how inorganic elements for life, or for a life, and is the presence of virus in a planet suggest that there is life in it? So if we find viruses on a planet, does that mean that perhaps life might have evolved?
Oh, these are great questions. Yeah, there's no simple answer to these. The answer is yes, I guess, is the short answer. I mean, if you go, there's lots of different theories about how life first started, and there's different theories. It's the old chicken and egg kind of theory. Is, what came first? Was it the virus or the cell? It's thought there was life started as an RNA world, with replicating RNA. And then it was eventually transformed into, a virus by giving it a coat to give it a little bit more protection. And so there's a lot of theories around, life starting as viruses, likely started as nucleic acids probably, but which then transferred to viruses. I was on a workshop many years ago. You can sort of look at, again, it's sort of looking at definitions of life, and you get sort of inorganic replication. So silica, I mentioned silica in the top there, where you can get forms of silica that can almost self-replicate. And if you got a combination of inorganic and organic compounds combining, which can form some type of rudimentary life. I was at a NASA workshop many years ago, actually, in Washington, DC, where we were looking at all these different questions. There's a whole report on it, actually, that if somebody's interested, I could dig it out. It's really sort of looking at the big question there was, if you were to put a space probe to Mars or something, how would you know what life looks like? And that was the question. That was the thesis of the actual workshop. What would you go looking for? I mean, my argument at the workshop, and why I was brought along, was, well, you should be looking for viruses. Find tools that you're going to find viruses, and if there's based on the Earth's example, wherever there's life, there's viruses, and we've got phylogenetic evidence that shows viruses have been there right from the start of life on this planet. So that's a sensible way of looking at it. When you're looking in extreme environments, there's now some evidence that the organic life or viruses in combination with silica is a way of sort of looking for life. And it's really about looking for some of these biosignatures that are going to help you identify what life is like. You see the cartoons of, life isn't going to be some kind of Martian that's going to come walking along with, elongated heads and everything. And it's an intelligent audience, that, but it's actually trying to figure out, well, what is it going to look like, and how are you going to be able to detect it, and what is the significance of it?
I've got about 1 million questions, but I do want to move on to careers. However, there are some questions here from one specific person who's got lots of quite intensely scientific questions, called Ernie. I also have a few on our YouTube from Zia, and these are looking at how viruses might have affected the development of us ourselves, animal cells, plant cells, thinking about the DNA machinery that might have come from viruses a long, long time ago, or potentially other things like the placenta of a human, how that might have viral origin. But I think we have touched on those things a little bit, and how viruses may have had a huge role in our development. So I'm just going to leave those as little topics for other people to look at. But I wanted to ask you about your coccolithophores and how they compare to other vascular plants like ferns and flowering plants, particularly when you're comparing different systems like involved in photosynthesis.
All right. Well, so the major difference between, I guess, microalgae and land plants is with microalgae, the single cell organisms, so they divide a little bit like bacteria. So they'll divide and then they degrade. So you don't get, and because they're single cell, they don't grow in the same way as a land plant would. So with a land plant, they're multicellular organisms, and you think of a tree where actually all the photosynthesis is going on in the leaves, but you're getting a buildup of biomass. There's a great analogy I used around, and I need to think about it now. It's called the moon board, where if you take all the trees on planet Earth and you create a plank of wood with them, and so that plank of wood would reach from Earth to the moon. Okay, so so you take all the lumber on the planet, and I can't remember the exact dimension of the plank of wood, but you take a plank of wood, and that's your moon board, they call it now. That's allowed to happen because you get a buildup of that biomass. In the ocean, with the photosynthesis that goes on, there's a moon board is created every day. Okay, but that doesn't build up. What happens is that is created once a day, and then it's eaten. Okay, so it doubles in size, and then that gets eaten or grazed by the zooplankton, the tiny microbial animals, or gets killed by viruses. So there's that production that's happening all the time, but there's still that sort of base core biomass that remains there, but it's doubling every day and then getting eaten every day.
That's an incredible scale. Yeah.
Yeah. So if that kind of analogy kind of makes sense, so that's a kind of fundamental difference, really.
Yeah, my mind's a little bit blown by that. That's amazing. Great analogy.
I was at somebody called Bob Anderson, who used to run the microalgae culture collection in the US. He came up with that concept many years ago. I loved it. I've used it in talks before. Yeah.
I wanted to move on for the last maybe 5 minutes, talk about your career and how I found it quite interesting how you were saying, oh, it turns out maybe I am an astrovirologist, and how if we did find life in space, suddenly that could be a shift in your career because of the history you've got and the expertise. So yeah, I just wondered if you could talk a little bit about your experience of your career developing over time, and maybe how your interests have changed as well.
Yeah, I mean, I talk. I mean, where I was brought up in Scotland, I was actually a fly fisherman, and so that's really I got interested in nature as a fly fisherman. So you're out fishing at a place called Loch Leven, which is about 30 miles north of Edinburgh, and now fishermen are brilliant at coming up for excuses for not catching fish, and usually it has to do with nature. and there's always something wrong. There's too sunny. There's too much algae in the water. There's always something going on. Of course, so that always sparked my interest, and I was always interested in nature and the reasons why we weren't catching fish. So that was probably the reason I initially got interested in science. My father was a fisheries manager on this loch, and we worked with. We had a fish farm actually that we used to sort of help stock the loch with fish. So I got interested in that whole process as well. When I was young, I wanted to be a fish farmer, and then when you start looking into it, and I sort of started reading into marine biology, and I thought marine biology sounds excellent, and this is an area I really want to get into. And I always thought, well, I'll always come back and be a fish farmer, maybe a salmon farmer, and in some of the coastal salmon farms or something like that. Of course, they're not environmentally friendly anymore, but anyway, that's when I got into marine biology, and I ended up getting really interested in microbiology. And it was interesting, to me. I remember seeing some lectures by an old lecturer, George Floodgate, his name was. He was interested in how microbiology, were responsible for the formation of oils. In fact, when you there's some microbial processes that go on there, but also was the buildup of phytoplankton over millennia, millions and millions of years. I mean, a lot of the oil that we get from that's pumped away up actually started as phytoplankton, really. So it's and so I got interested in a lot of these processes, particularly geological processes. That's why I like using the White Cliffs of Dover as an example, because you sort of combining biology and microbiology and geology and that kind of historical time and planetary formation and everything. So it was how everything was interconnected that I really enjoyed. And then eventually so I knew I wanted to do microbiology of some sort. And at when I was looking for a PhD, there was the topic of viruses came up. It was brand new. I thought that it'd be a great challenge. There's nobody really knows very much about it. So I was very lucky in that when I started my PhD, which I went to the University of Warwick to do my PhD. I was there right at the start, and that's why I know a lot of the really well-known environmental virologists, because a lot of us started around about the same time, and that was about 30 years ago. But I always probably the real reason I got into marine biology is because the field trips were always brilliant, and so it was just part of it. You got to go out and do interesting stuff. I used to, certainly when I was younger, I was at sea, 3-4 times a year. So I've been in research cruises. Well, we don't call them cruises now because it makes it sound as if it's something really dossy, but it's expeditions, research expeditions we used to go on. I've kind of been all over the world in different expeditions. So with the focus still on marine microbiology, I spent quite a bit of time working in Bergen as well. So some of the coccolithophore work, that's how I really got started on that in Bergen in Norway. And so, yeah, just expanded from there. We've sort of built up a research group doing all sorts of different marine virology. I've been involved with coral biology as well. So sort of looking at coral reefs and the coral reef bleaching. And we kind of thought, well, viruses are going to be involved in that somehow. So we did a lot of work on that over the years. I've been off the coast looking at some of the reefs off the coast of Africa with some of that work. I've worked, as the video showed, the radio lab video. I ended up working in the United States, in the state of Maine, which is as far northeast as you can go in the US, right? It's a beautiful part of the world, at the Bigelow Laboratory for Ocean Sciences, and there I ended up taking charge of the culture collection, which is one of the largest microalgae culture collections in the world. And there it was, I always described it as a kid in a candy store. You got all these phytoplankton that you can play with, and we were just isolating lots of different viruses and doing the characterization and just really finding out about viruses. And one of the great things about virology, and that's why I've stuck at it for 30 years, is I know I could pick a system that nobody's worked on. We have a project that's just starting up. We're looking at viruses that infect fungi, marine fungi. So it's really, there's one of the research fellows at the Marine Virological Association, Michael Cunliffe, has a big European research grant that's looking at marine fungi, and that's what, the chatting with people in the corridors and at coffee time, and I always ask the question, what about viruses? And we think, right. And there's one thing I'll say to all your students: if you really want to annoy your teachers in any kind of biology, I just ask the question, so how are viruses involved in this process? Because we know that viruses, and usually there isn't. Sometimes there's an answer, but quite often we don't know. So I've always been pretty passionate about virology, because they're involved in any kind of biology, and even, you can relate viruses to a huge number of different scientific topics. So it's just that there's so many different routes that you can go. But my whole pathway has been really, I started in Scotland. I did my undergraduate in Wales, in Bangor in North Wales. Then I had my PhD in England in University of Warwick. I came down to Plymouth, where I was a postdoc. I ran a research group, and then I went over to the United States and so I worked there for about 8 years, and eventually came back. Now I run the building you see behind me is the Marine Biological Association. My office is right in the corner, and I look out over Plymouth Sound, so about where I'm sitting. So I have one of the best views in Plymouth. So it's a nice place. Come and visit us.
Yeah, that sounds amazing. There's so many things that I'd love to continue and talk about, and we have to wrap it up now. But just the importance of phytoplankton, the point that you made, which I think is fantastic, which it's not either or. It's not just bacteria or just viruses. They're working together, and that we're not getting the whole picture, the whole story. But great distinctions. Thank you so much for being with us. What an incredibly interesting talk. And field. And I absolutely agree with you as also a field scientist, as to expeditions, traveling the world. It's an amazing career to be on, because you get to interact with so many different people from around the world and see so many different amazing places when you're working in these types of fields. So all wonderful, fantastic. I want to thank you, Professor Wilson, for being with us, and the MBA for allowing us to have some of your time today. Thank you, Joe, from the Linnean. We are going to continue next week, again, with the Burlington House Lunchtime Scientist Astrobiology Talks. So who do we have next week, Joe?
Yes, we have Emma Ryan, don't we?
Not Emma Ryan. We have Dr. Alex Price. And
Emma Ryan's the week after.
Yeah, this is great from Professor Wilson, because he touched on, and thank you again also, Professor, for contacting Ken Stedman who is, you term him as the astrovirologist, showing some images of him working in extreme environments. So our next speaker is going to talk about working in extreme environments and doing analog research in connection to astrobiology. So all really fantastic stuff. Thank you for being a part of this. We really appreciate it. And we'll see you all next week. Thank you. Bye-bye.
Thank you.
Thanks, Willie.
Appreciate the invite, and good luck for the rest of the series.
Thank you very much.
Thank you very much.